and welcome to the Sleep Science Podcast. I'm Penny Lewis, a sleep scientist and the presenter of this program. In this show, we talk about all things related to sleep, from dreaming and sleepwalking to what sleep does for our brain and body and how we can get more out of our sleep. This program is a production of the NAPS Sleep Lab at Cardiff University. You can find more information about the guests, the podcast, and our sleep research at Cardiff on the Sleep Science Podcast webpage. If you enjoy the show, please don't hesitate to follow us or like us on Twitter. In this episode, I'll be talking to Gina Poe, a professor in neuroscience at UCLA. She's one of the pioneers of rodent work, researching the connection between sleep and memory, with a particular focus on REM sleep. We talk about the importance of REM sleep for memory consolidation, ways this has been studied, and the potential of sleep manipulations to reduce some forms of anxiety, including PTSD. Finally, we talk about Gina's groundbreaking findings on local sleep and how these may lead to changes in our approach to sleep research as a whole. So firstly, thank you very much for agreeing to do this, Gina. Could you tell us about some of the work that you've done on REM sleep and on memory replay in REM sleep?
When I started research back in 1991, a long time ago, we of course knew what REM sleep was. We knew it was a really vulnerable state, and we knew that it was important for learning and for memory, but we didn't know how. We had no idea what the mechanisms were in REM sleep that were important for learning and memory. A few of the features that were unique and interesting were that during REM sleep, the hippocampus, which is really important for learning and memory, was constantly in a state of theta. And theta is a brainwave signal from 4 or 5 hertz, 5 cycles per second, to about 10 cycles per second. And we knew at the time that theta was really important during waking to being able to learn something new. And if you didn't have it at the time that you were trying to learn something, then you wouldn't learn it really well at all. And that theta went away in slow wave sleep. Theta was gone when you're grooming or doing kind of any automatic activity that doesn't involve concentrating and putting together new associations. But it was really important during kind of an associative learning and memory task where the hippocampus is known to be important. So it was really interesting that theta was constant and really big during REM sleep, but still people didn't really even know why theta was important for learning. So theta during REM sleep was also a complete mystery. But right around the time that I was about to finish my graduate research and start my postdoc, I was going to a lab that was doing research on recording the hippocampus in learning animals. And I wanted to record during sleep. And I had just heard a lecture by John Lissman that told us that if he stimulated the hippocampus at the peaks of theta, which is when all the cells are most likely to respond to that incoming input, that he would get really nice potentiation of synapses, which is kind of thought to be a building block for strengthening circuits that are involved in encoding memories. So if he stimulated at the peaks of theta, he got long-term potentiation. And really interestingly, if he stimulated the troughs of theta, he would get depotentiation or weakening of synapses. And just around the time that he was publishing that, or a few years before that, Francis Crick had published a paper showing that not showing that anything. Actually, the paper was just a hypothesis that maybe REM sleep is for learning, because really what's important during REM sleep is you're erasing all of those things that you learned during the day that aren't important to remember. So it's an important time to clear the brain of all the nonsense that you don't need to remember long term. And so it was just a one-page, hypothesis. And maybe REM sleep is for clearing the brain. But it didn't really jive well with those who had been doing research on REM sleep and memory because, they knew that it was important for forming new synapses. So I knew I was going to this lab where I'd be able to record from hippocampal neurons during REM sleep theta and see whether or not cells fire at the theta peaks or the theta troughs to see if REM sleep was for remembering or for forgetting. So I was really excited to go and do that project. And I was able to see that it was both. And It depended on the strength of the memory, the length of time that memory had been in the hippocampus, and whether that memory had been transferred already to the cortex.
Can you say a little bit about what memory replay actually is and how you measured it?
Yeah. One of the really exciting things that was discovered just before I'd be able to study the mechanism for REM sleep and memory was that during sleep, memories seemed to be replayed. And what that meant was that the neural activity that was involved in forming a memory in the first place was reactivated again during sleep. And this paper by Matt Wilson and Bruce McNaughton showed that animals who were running around on a maze and cells that were active at different parts of the maze were reactivated again during sleep after this maze and were replayed in fast forward motion. So for example, if it took a minute for the rat to run around this maze, it took maybe 200 milliseconds to run around half of the maze in this replay event. During slow wave sleep, when it's known that if you wake people up out of slow wave sleep, they'll reply that they weren't conscious of anything, they weren't dreaming. So even though these were rats and we couldn't ask them, we could also, hypothesize that they weren't actively dreaming about it. So one of my questions was, are they replaying during REM sleep when you do wake up people and they say, yes, I was dreaming about X, Y, and Z? And one thing that Matt Wilson talked about and published and saw in my data was that the replay during REM sleep, yes, things are replayed, but they're replayed in real time, which was really interesting. So that it's as though the animal were experiencing the same maze in the same amount of time as they took to navigate it in wakefulness. So that's what replay is. And the reason why it's exciting is because it's thought that, rehearsal is important for continuing to strengthen synapses. So this is a time you could rehearse these things without having all the noisy input from the world interfering with it.
And so the way you actually index it is by looking for particular sequences of cells firing that had fired during wake and then fire again during sleep, isn't it?
Yes, because in fact, there are lots of other orders of activity and firing patterns, but we don't know unless the animal has done that during a waking behavior, whether those random, what we would think are random, are really random orders of activity, or whether it is a replay of something that we just didn't see during wakefulness. So it could be that there is no noise in the brain at all. It all has to do with something, but we can't know that unless we record every cell, every moment of an animal's experience to know, what that cell is associated with trying to do.
And are these particular special cells, or are they just any cells?
So the cells in the hippocampus are special because they are considered to be the highest association cortex because they are connected with every other area of the neocortex. So areas that are involved in decision-making, areas that are involved with listening, and areas that are involved with speaking and seeing and feeling, all of those areas connect to this hippocampus, which is down deep in the temporal lobe, so beneath the ears. And what it can do and what it's known to do is to make associations between all these different modalities and connect them in ways that take a long time to do in the cortex. For example, the typical Pavlovian dog learning the association between the sound of a bell and the presentation of food. There are two areas of the brain that aren't normally well connected. You don't think of bells and food together, but because both listening to the bell and smelling the food and eating the food are all connected to the hippocampus, and the hippocampus is all well interconnected with each other, it's easy to strengthen synapses between these areas in the hippocampus. We think now, and there's good evidence for it, that the hippocampus then trains these different areas of the brain to reconnect during sleep and strengthen them additionally in these areas of the brain. And it takes a while, it takes about a week, but they can be strengthened so that then you can remember these things and these areas of the brain can fire together without the hippocampus present. So after about a week's time, you can lesion the hippocampus and still retain that memory. So are these special cells? Yes. Especially in the fact that the way they're connected with the rest of the brain.
Okay. And then I guess the obvious question would be then, how does this replay impact on memory consolidation. And you mentioned that in REM, it impacts both on kind of getting rid of unwanted memories and strengthening other memories. Can you talk about that?
So one of the interesting things is that when we're awake and learning new things, we actually cannot unlearn anything that we've learned before. So for example, if you learn that Santa Claus, and let me see if I can say this without disappointing anyone. So when you learn new facts about who Santa Claus is, you have to loosen your schema so to fit it into your old schema. So Santa Claus is not just a jolly old man who lives in the North Pole, but he was a real man who made toys a long, long time ago for kids in the middle of winter when they're going crazy with cabin fever. He would distribute them. So to be able to fit that new piece of information in with the old, you have to sort of open up your network of all the identities of Santa Claus. And in order to do that, you have to loosen some of the ties. And that can only happen in REM sleep. And the reason why it can only happen in REM sleep is that's the only time that a couple of neurotransmitter systems, chemicals in the brain that influence how your brain works, are off. And the reason why it's important for those neurotransmitter systems to be off during REM sleep is because these neurotransmitters only allow the strengthening of connections. It doesn't allow the weakening of connections. So REM sleep is the only time you can sort of loosen that network to reassociate and incorporate new pieces of information, new facts, new insights. And that's what our research showed.
So you mean essentially updating schemas?
Yes.
How did your research show that this can only happen in REM?
Okay, well, our research actually showed that the neurons in the hippocampus are only firing at the troughs of theta when John Lissman found you could get depotentiation or weakening of synapses. That could only happen in REM sleep. Neurons don't normally fire at the troughs of theta during wakefulness, but even if they did, in the presence of these neurotransmitters that are only off in REM sleep, it couldn't loosen those synapses. So it was in part observation that cells only fired at the troughs and knowledge that these neurotransmitters were only off during REM sleep that we put together to say the effects would be to weaken and loosen these schemas so that you can update them with new information.
So you needed the replay to happen at a time when things could be depotentiated to update the schemas. I see.
Yes. And actually something new relative to that first discovery is that there's another time when these neurotransmitter systems are off, and that is during the transition to REM, which in humans is called stage 2 sleep, when you have lots of sleep spindles. And these neurotransmitters are off for a couple of seconds before every sleep spindle. So it's really exciting to think that at this unique time, sleep spindles are these 10 to 15 hertz large waves that you see both in the hippocampus and the cortex. And it's a time when the neocortex and the hippocampus are kind of uniquely connected. They're in sync with each other. Much more than they are during any other time of sleep. So it's really cool to think that at the beginning of these sleep spindles, when these neurotransmitter systems have been off, you could actually loosen the schema that are encoded in the neocortex. Then at the end of the spindle, actually these neurotransmitter systems turn back on and they allow you to really easily strengthen new synapses. That's when you can incorporate the new information. So the beginning of the spindle could be a time when you loosen the schema, and the end of the schema could be a time when you've sewn in the new piece of information.
Oh, that is so interesting. So I didn't know that about sleep spindles. Can you tell me more? Tell me about the data behind that.
Yeah. So what we're looking for right now in our replay data during these sleep spindles is to see whether or not memories or schema are replayed in a backwards mode at the beginning of sleep spindles. And that would be really cool because played backward, there's something called spike timing dependent plasticity that would, in combination with the absence of these neurotransmitters, which are norepinephrine and serotonin, it would allow the weakening of synapses. And then what we hope to see is at the end of spindles, when these neurotransmitter systems are back on, you would be able to see forwards replay which would include replay of the new pieces of information. So is backwards replay not include the new information, and forward replay at the end include the new information? And so far we've had a couple of examples at least that we can see of that, and now we're systematically going through all the data to see if that's true.
Does that link in at all with your recent current biology paper about norepinephrine and spindles?
Yes, it does. So we replicated old data showing that these neurotransmitter systems, especially the norepinephrine system, is off before every sleep spindle, and that it fires in a burst right in the middle of the sleep spindle and very quickly after the sleep spindle ends. So we have seen that if we disrupt sleep spindles by turning on the norepinephrine system too soon, so in other words, we curtail all sleep spindles if we make the locus coeruleus, which delivers norepinephrine to the forebrain, if we make it fire 2 times per second, so we never let it go silent, we actually prematurely end spindles. And we definitely find that animals can't update their schema at all with new information. And now we're starting to look at the replay that happens during these curtailed spindles. Are they not ordered? Are they not able to loosen the schema and weaken the synapses and then incorporate new information? It is particularly the longer spindles that are curtailed by our stimulation of this locus coeruleus that keeps norepinephrine in the brain.
So let me check if I got that right. So in that paper, the main point was essentially suppressing the spindles by stimulating the locus coeruleus, which meant that it produces norepinephrine, which it should not do at that time. And then when you did that, kind of repeatedly, animals were unable to consolidate their memories.
Yeah,
so they didn't update.
That's right. They did not update. And the task that we gave them, it was an updating task. We had them running a maze that was very familiar to them. They knew where all the food was. They only checked those boxes which contained the food, and they ignored all the rest. And then after running this familiar maze, we switched the places that the food was. So they had to learn to start ignoring the old boxes and checking only the new boxes. And that's updating an old schema, because all of this, the whole maze was in a very familiar room. They're running at a familiar time. Everything was the same. The rules of the task were the same. It was just the places which contained food were changed. And what happened is that animals that we didn't stimulate with norepinephrine, so we allowed the norepinephrine system to go silent when it should, for example, before every sleep spindle, those animals were able to learn the new schema fine and update their schema. And those that we disrupted and left norepinephrine on were not able to update it. And so they just got confused and started checking all the boxes.
So that was the current biology paper, which is really exciting. But then the new work that you're doing to build on that, you say that you're particularly looking at the longer spindles and looking for differences between what's getting replayed at the beginning and the end of the spindles, with the idea that if they need to update some information, they should be replaying the old version at the beginning of the spindle and then hopefully replaying the new version at the end of the spindle.
Yeah, that's what we're looking for. Exactly.
And you have found examples of that.
We have definitely found examples of that. So now we're going through systematically and seeing if that's true more than would be due to just chance.
Is that using the same kind of maze task? So what they have to do is update a schema for the maze with some new boxes that would have food in.
Yes, exactly. Yeah.
Do you have ideas about how many times, how many spindles this would have to happen in?
You know, I don't think it would take much, because with that John Lissman paper, they were able to obtain depotentiation with one burst at theta troughs during REM sleep. So I think if the conditions are just right, it wouldn't take but a few hundred milliseconds to update a schema. One spindle might be enough to do it. Now, it depends, I guess, probably on how complex the memory is and how many synapses in the network need to be changed. So I would imagine a very complex new memory might take more than one spindle, but I still don't think it would take very long.
So is the idea the old memory, the original memory, would get replayed in the troughs at the beginning of the spindle, and that would allow it to be kind of deconstructed somewhat through this kind of depotentiation. And then somehow the new information would get inserted, and then the new memory would be played in the peaks, which allows for potentiation and strengthening of that new memory.
Yeah, that could be. Certainly in REM sleep, troughs versus peak. I'm not sure in sleep spindles whether that's true, but it's quite possible. And we haven't done that level of analysis, whether they're played in the troughs versus the peaks of the spindle, but it's perfectly possible. All it would require, though, is that the spindle's so fast that the memories would just need to be replayed in a backwards order. So say you're running from A to Z in that order. If the memory is played from Z to A in a quick succession, then that would be enough to cause heterosynaptic depotentiation through spike timing dependent plasticity if you don't have norepinephrine present. So all you need is backwards order to open it up.
Can you explain how that works? Heterosynaptic plasticity.
Yeah. So heterosynaptic plasticity, say presynaptic and postsynaptic side. So there's someone knocking at the door. That's the presynaptic side. And then someone answers the door. That's the postsynaptic side. And if someone answers the door and there's nobody there, then they are less likely to open the door the next time someone knocks, right? It's kind of like this ding dong ditch, I guess. You just say, okay, nobody's really there, and you start ignoring it, right? And the same if someone knocks at the door, the presynaptic side is active and the postsynaptic side does not respond, then the person coming to the door is less likely to come to the door again the next time because no one's home, no one's answering them. So basically the door is the synapse, and the synapse gets weakened if someone opens the door and no one's knocking, and if someone knocks and no one opens the door. So both sides are heterosynaptic. It's called heterosynaptic because the only one side is playing the game.
So basically, if the two sides don't work together in the way that they should,
yes,
then both sides get depotentiated.
Yeah.
Okay. And so if it gets replayed backward, why would it allow depotentiation?
On an intracellular level, what it means is that, so let me see, let's start with someone knocking and no one answering. So this is if there's a lot of glutamate released, for example, by excitatory neurotransmitter, the knocking gets released, but the postsynaptic side isn't able to fire an action potential in response to that. So the receptors still get occupied by glutamate and lets a little bit of sodium in, but not enough to fire a whole action potential. And that little bit of depolarization also lets a little bit of calcium in, or lets a little bit of internal calcium free from their stores. And just a little bit of calcium sets in motion a cascade of intracellular events that dephosphorylates things that were initially phosphorylated in potentiation. Sorry, I'm getting into the weeds here. Essentially, what I'm trying to say is it basically unlocks and takes down the receptors. So it depotentiates. So that's if the presynaptic side is releasing glutamate and the postsynaptic side is not responding. Similarly, the postsynaptic side is activated, say, by something else, but not the synapse that's there. So say the postsynaptic side is firing because somewhere else in its dendritic tree, it's got it excited. And so the postsynaptic cell is firing, but the presynaptic side is not the part that's exciting it. So then you also get just a little bit of depolarization, a little bit of calcium in, and a little bit of calcium does the same thing there also, which is just takes down the receptors and starts the depotentiation sequence of events. Wonderfully, because not every synapse is active every time a cell is firing an action potential, during waking with norepinephrine present, and even during slow wave sleep when norepinephrine is present, you don't get depotentiation because it blocks that. Norepinephrine acts at a beta receptor to lock down memories and keep synapses the same as they were before. It also helps long-term potentiation if both pre- and postsynaptic sides are there. So it pushes everything towards long-term potentiation or strengthening, or at least preserving the synapses that you have there. So that if you think a new thought, you're not going to erase everything else.
I think I understand now. So that's why it's important that you have this inhibition of norepinephrine at the beginning of the spindle, because that's when you think that this kind of deconstruction, this flexibility can happen. Yes. And if you artificially boost the norepinephrine at that point, then it can't happen, essentially.
That's right.
So you can't have plasticity. So I know that you have some really interesting work around emotional memories and PTSD. Can you tell that story?
Sure, yeah. And I think it builds on what we've already talked about because it's already known that people with PTSD have a heightened noradrenergic system. So norepinephrine is higher in the central nervous system of people with PTSD when you measure it while they're asleep or when they first wake up after a night's sleep. So that's the time when it's most differentiated because wee hours of the morning REM sleep, when we get most of our REM sleep, is the time when that noradrenergic system is supposed to be off. So that's the time when norepinephrine is lowest in our brain normally. But people with PTSD don't have that dip, that early morning dip. And so it's our hypothesis that they're getting REM sleep because, of course, they do report dreams, but it's not REM sleep absent norepinephrine. And again, what I said that norepinephrine does is it helps us strengthen synapses, but doesn't allow us to weaken them. So REM sleep and that transition to REM sleep stage 2 is malfunctioning in people with PTSD. They cannot loosen their schema that was formed around the traumatic memory. And so new pieces of information are into it, like, yes, that was then, but this is now. Now I'm safe. That person is gone. I'm in a safe place, all of that. So they can't sew in that new context into that old traumatic memory because they can never loosen that schema. So with rats, we expose them to a trauma, and then we can see what happens to their locus coeruleus during REM sleep. And if they get PTSD, not all of them will, just like people, not all people will get PTSD, but those rats that do get PTSD-like symptoms.
How do you measure PTSD symptoms in a rat?
Okay, so one of the things that people have with PTSD is they aren't able to encode new context in with an old fearful memory, and they can't extinguish that old fearful memory when in the light of new context, right? And the same thing with rats. So what we do is we pair, for example, a tone and a shock in a particular context, and they learn to fear the tone and the context. And then we bring them into a different context and play the same tone. And we do that enough times, like 30 times over an hour and a half, but we never shock them in that new context. And what the normal rats can learn is, and actually even rats that have a PTSD phenotype, they can still learn that, okay, now I'm not going to get shocked. You know, it's been 30 minutes, it's been an hour, and I've never gotten shocked. So they start relaxing and just wandering around and they stop freezing. But the next day, when you bring them back into that same safety context, those animals that have a PTSD phenotype can't remember that they're safe there. They haven't consolidated the safety parts of that context. And so they still freeze. And that's similar to people with PTSD who will continue to have all the fear responses. And when a cue is presented, they associate with the trauma, despite the fact that it's been years.
You started to say before what you did to these rats to kind of produce the PTSD symptoms.
Yeah. Right. So we just exposed them to a trauma, and it really doesn't even matter what. It's really interesting. It doesn't seem to matter if the trauma that we exposed them to is in any way related to this context of a tone and a shock. It just sets a maladaptive sleep in them so that forever after they can't loosen their schema and update it. And this happens in both fearful kinds of memories and memories that are not fearful. So it would be really interesting to test in people with PTSD if their sleep turns maladaptive and they also then have difficulty updating their schema in all kinds of contexts, both fearful and non-fearful. So that's our hypothesis, is that what happens with these rats is that their REM sleep occurs in the presence of norepinephrine. The locus coeruleus remains firing and becomes maladaptive, and that sets up a positive feedback loop of continuing to fear during the day and continuing to fire their locus coeruleus during the night when they're asleep, and never allows that loosening of schema.
So what you actually did was traumatize the rats. And then, am I right in thinking that you then watched their behavior after the trauma? And some of the rats stayed awake for some time after the trauma, and other rats went to sleep. I mean, they were given the opportunity to sleep. Some of them did go into REM sleep quite fast. Those who went into REM sleep with this kind of heightened level of norepinephrine then subsequently develop the PTSD. Is that right?
Yeah. Yes. Good memory. So that's exactly what happened. So an adaptive response after the traumatic event was insomnia. So rats that stayed awake for longer periods of trying to figure it out or doing whatever they did. And if they were able to stay awake and not have a lot of REM sleep for the first 6 or 7 hours, those rats all showed resilience. A week later, when we were testing them for their PTSD phenotype, they were all normal. So it appears that REM sleep, or a lot of REM sleep, in the first 6 or 7 hours while the locus coeruleus is still responding to that stressor.
And it's important to say here, I think, that normally the locus coeruleus would not fire in REM sleep, right? So that's a time when there should not be any norepinephrine. And so if it's firing because they were stressed and then they go into REM sleep nevertheless, then that REM sleep seems to be maladaptive.
Yes, that's right. It becomes maladaptive, and then it becomes a positive feedback cycle that I think can be broken. And that's the exciting part of this research, is that we can break that cycle in rats by just giving them one opportunity to sleep without that heightened noradrenergic system. So the way we're testing that is that week later, when we're exposing them to tones and shocks and then tones without shocks, if we give them propranolol, which blocks the beta noradrenergic receptor, they will be able to consolidate that extinction memory, that tones without shocks, safety context, and thereafter they do not display the PTSD phenotype. So the reason why we did this experiment in the first place is because quite a long time ago, I guess it was in the 90s, Jim McGaugh at UC Irvine gave people one big pill of propranolol after having them write down their traumatic memory in detail. So he had them recall that traumatic memory, but in the context of his safe laboratory. And then he gave them a pill of propranolol and sent them home. He did not measure whether they went to sleep or not. He wasn't thinking about sleep at all. He was just thinking about the consolidation of that context memory. And in fact, what he found a week later when he brought them back and had them write that traumatic memory again is they were able to recall it without all of the emotionality that they had the first time. So without the palm sweating, without the heart racing. And so that one pill just and sending them home was enough. After that, people got really excited about propranolol, and he tried it in 7 different clinical trials. And unfortunately, half of the clinical trials worked and half of them didn't at all, or even seemed to make things worse. And so people have dropped it, and they're not looking at propranolol at all anymore. And the reason why I think that's true is because, again, none of these studies take sleep into account. And so I think if propranolol wore off before they went to sleep, then they weren't able to have that peaceful sleep that they should have had, a normal sleep that they should have had. Or if they gave propranolol before they had the extinction exposure therapy, they weren't able to learn the new context because it blocked their ability to do the associative learning in the first place. So, of course, they can't consolidate it.
So in these studies, has anyone reviewed those studies to look at the timing of the propranolol? And
no.
Are you going to do that?
Yes. Well, I'm doing it in rats. I'm doing the new experiment in rats. I'm timing propranolol to 3 different times and giving saline to other animals. And it's been extremely effective if you do it at the right time, and extremely ineffective, or even making things worse, if you give it at the wrong time in these rats. So I can totally see, in relation to timing of sleep, how it could have been that these studies that didn't take sleep into account could have been so mixed in their results.
You're making me wonder if we should do that in humans. Let's wait and see what you find in your study, and then tell me, and we could follow that up.
I can send you some graphs. It's pretty startling. So we haven't published it yet, because what we're doing is, in this last 4 rats, is recording sleep. And before, we weren't recording sleep. We were just studying their behavior and giving the propranolol at the right time in relation to sleep. So now we have to confirm, in fact, that their sleep is peaceful versus not, and then we'll publish the paper. I feel like it's so important. It's almost as though it's an essential study, because PTSD is such a huge problem. In the United States alone, I think they say 22 service members a day commit suicide. And that's a lot of lives lost.
Yeah, that's a lot.
PTSD.
So there is a human study relevant to this that I wanted to ask if you would comment on. So have you seen this study from Oos van Sommeren where they had people who had either disrupted REM or normal REM, and they looked at their emotional memory processing and they applied TMR to get them to reactivate these memories. They used odor TMR, and they found this startling difference between the people with the disrupted REM and the normal REM. He certainly cites your ideas as the explanation for his data.
Yeah, I think that this is just so exciting. I couldn't believe, first of all, how inventive the studies were and how perfectly they align with ours. It's a beautiful study. And the reason why I think they get such disrupted REM is because the locus coeruleus is firing. And when the locus coeruleus is firing, it sort of sets up the whole system to wake up more easily, which is another reason why probably people with PTSD remember their nightmares so readily, is because they probably are awakening from their nightmares a lot more often than people normally awaken from REM sleep. Yeah, so I mean, it would be great if he could record from the locus coeruleus of people, but unfortunately it's way deep down in the brainstem and you can't get it with anything that we normally use to record sleep in humans.
Yeah, but still, his data are startlingly consistent with your ideas.
It is really startlingly consistent. And I'm thinking of ways to assess the locus coeruleus activity in people who are asleep without being invasive. And I can think of a couple ways that would only take a little bit of engineering to get. I really want to invent this device that would be able to assess this. It would be great to get someone who's been exposed to a trauma and be able to prevent them from having this maladaptive sleep in the first place by assessing the amount of locus coeruleus activity. And if there's too much and they're not ready to go into REM sleep, it could either, through automatic feedback, deliver this propranolol drug so that it arrives in time to allow their sleep to be adaptive, or it could wake them up and prevent them from having this maladaptive sleep and continue that until their locus coeruleus finally calms down. Because something that we didn't really talk about very much is that those animals that had a lot of insomnia, they did finally go into REM sleep and had quite a lot of it, but it was after the period of time when the locus coeruleus would have normally calmed down after the stressor.
And then their prognosis, their outcome was much better, right?
Oh, yeah.
I was just going to ask about your work on local sleep, because I think that's also really important.
Oh, it was a long time ago now, wow, almost 20 years ago, that I noticed when we were recording from the hippocampus and the cortex at the same time, that often the hippocampus would show that it was in REM sleep. It would go into that high theta mode before the neocortex. The neocortex would still be in the non-REM sleep mode. And so we kind of thought, hey, that's weird. What's that all about? And so it took us a little while to figure out how to analyze it, but we found that the hippocampus was indeed going into REM sleep many seconds, sometimes 20 seconds or more, 40 seconds before the cortex would finally transition into REM sleep. We wondered whether this happened more often when an animal was learning, and the hippocampus would therefore have higher demand for REM sleep. And we found that that was also true, that the hippocampus spent more time in REM sleep than the cortex, just after the animal had learned something really new that needed to be consolidated. This was really cool to me because people had shown that different areas of the neocortex could be showing high volume delta waves, while other areas of the neocortex were not in that high volume delta sleep, but was still in non-REM sleep. And people had shown in seals and marine mammals that one hemisphere could be asleep while the other hemisphere was awake. And people had shown that in birds as well. But no one had shown whole areas of the brain like the hippocampus versus neocortex in different sleep states at the same time. And so that to me was super exciting.
Super exciting and really important, I think, because it changes the way we look at sleep, in fact. I mean, certainly when we read that paper of yours in my group journal club, we all kind of went, oh no. Because this means that all of the work that we've been doing, where we're using cortical electrodes to determine the sleep stage, and then we're inferring things about what's happening across the whole brain to memory processing, and we're assuming that it's all in the same stage, it's just wrong.
Yeah. I'm sorry.
But I would say it's like anything else in science. Of course, it's always much more complicated than you think.
Yeah.
So we haven't despaired. I mean, we continue to record EEG from the electrodes and determine sleep stages, but at least we're aware now that we're just scratching the surface.
And they are in the same sleep stage most of the time. It's just at the transitions, especially when they're not both transitioning at the same time. But it was strange enough that, in fact, sometimes, it didn't happen that often, but sometimes the rat's hippocampus would go into REM sleep for 20 seconds or so and then come back out into non-REM sleep, and the cortex never transitioned. So it was like a whole REM sleep episode happened in the hippocampus where the cortex just never followed. What this meant to me was that those studies which deprive animals of REM sleep based on EEG weren't necessarily depriving the hippocampus of REM sleep.
Although those studies do tend to show really dramatic impacts on memory and on physiology in many ways. So even if the hippocampus is sneakily getting a little bit of REM sleep in there, it's obviously not enough to prevent.
It's not enough.
Prevent those drastic symptoms of not enough REM sleep, basically.
Absolutely. You're absolutely right. Yeah. Which makes me wonder what's really happening to brainstem areas, which are supposed to be controlling aspects of REM sleep, like the lack of locus coeruleus activity and dorsal raphe serotonergic activity. Are they in REM when the hippocampus is in REM, or are they in REM when the cortex is in REM? Or are there subgroups within these nuclei that project specifically to the hippocampus and to the neocortex then are in the REM mode, right? There's so many questions left to answer.
Yeah, I mean, obviously, the deeper you go, the more fine-grained you get, the more complex it's going to get. But the initial point, I think, that your paper made really nicely was that it's not homogeneous. It is completely heterogeneous. I think that's a really great note to finish on. Thank you so much, Gina. It's just so interesting, all of your work. I could continue listening for hours, I think, but I have to let you go.
Well, thank you, Penny. And your work also is extremely interesting. Someone should interview you.
Take care.
Thank you, Penny. It was great talking to you.
It was really great. Bye-bye. You've been listening to the Sleep Science Podcast, a production of the NAP Sleep Lab at Cardiff University. I'm Penny Lewis. My guest today was Gina Poe, and our producer was Eniko Simo. If you've enjoyed the show, please consider subscribing to the podcast or liking us on Twitter. If you have questions or comments, please get in touch via sleepsciencepodcast, all one word, @gmail.com. Thanks for listening, and until next time, sleep well.